Mark, you have a lot of experience with the Lung Cancer Mutation Consortium. It's still only 4 to 8% of patients.
And that's an overestimate.
And
I mean, about
that are ALK positive.
Oh, well, it was 8% actually in the LCMC. It was a
Still
little
a
higher than
minority.
people. But you know,
Single-digit minority.
but I just wanted to take one quick step back here and talk about crizotinib. It really was a game changer.
Yeah.
We can't forget that. I think Mark brought up some very, very good points. By the way, that MET trial is still open.
Yeah.
It's still open. But just a couple of cool things about it. The first is, I don't think a lot of people didn't believe the EGFR story. They thought it was hokum, maybe because I said it was true. But they needed another target that meant something to people, and this was it. The second thing was, because it was so effective and so obvious to everybody, it totally flipped the drug approval process on its head. Think about it, a first-in-man drug approved for anybody with an ALK fusion based on 256 patients, a handful that got it first line.
On a response rate.
A handful. But
Prior to
nobody,
the PFS
but
data.
nobody doubted that this drug was helpful. Docs, patients, regulators, it was true. The other cool thing was, a lot of these patients, because they were never smoking and women, they had erlotinib. And of course, this erlotinib is ineffective against ALK. And these patients tell you a beautiful testimony for if you don't have the target, despite the fact you have a pro-trial, the target therapy works. They taught us to treat beyond progression. Again, go back into that CRIZ stuff. You did not stop the CRIZ when you had resist progression. Docs in that initial trial were allowed to continue people on, and that's the way it went.
Particularly for oligoprogression, where patients were still having a clinical benefit but had one site or perhaps a couple sites that were progressing that could be addressed separately.
And it's been changed and now part of NCCN guidelines. It also works in ROS1. You mentioned the multi-kinase. It works really well if you're MET amplified. It works really well in ROS1. I think many of you saw that data that El Shaw presented as well. So it's really, really been a game changer. One thing we don't know about this, though, is the real mechanisms of resistance. It's not as clear-cut and simple as it was with EGFR. 60% of people with an EGFR mutation under the stress of TKI develop a second-site mutation. There have been case reports of second-site mutations with these ALK drugs, and a lot of different things. So it's probably a different mechanism, and these just might be better drugs, more potent drugs. And that may be the benefit of them. I'm not sure that they work better in a resistance setting or not. I mean, the jury's still out on that, because we don't have a good scientific way of documenting a common pattern of required resistance. But all these are really good drugs. They appear to be safe. They appear to work in crizotinib failures. And at least the crizotinib has the breakthrough designation, and I bet we're going to see it this year.
So Corey, you asked me,
how do you develop?
So I mean, obviously we have a standard out there. It's crizotinib, and it's got good data associated with it, and this drug works. The strategies could be looking at crizotinib failures. I think that's a complicated strategy, as Mark mentioned. There's seems to be a bit more heterogeneity in the mechanisms of resistance to these drugs, and if you don't do dedicated rebiopsy studies and re-look at the genomic part of it, you could be misled based upon what your mechanism may be, or go head-to-head against crizotinib,
which is what's
happening with the LDK drug. And so we'll see if we replace crizotinib. You mentioned the GI toxicity. I think that's clearly dose-related. I'm not quite sure you need to use an MTD dose in a targeted population. So use the lower dose rather than the higher dose in a targeted population.
Although those of us who've had patients who've received both crizotinib and this LDK, the crizotinib compound, it seems even perhaps with dose reduction, you do see a bit more GI toxicity with.
We're still not sure about the right dose. And please remember too, crizotinib, dose-limiting toxicity, GI. Why is it given BID? Cut down on the GI toxicity. And those of us that have seen toxicity with criz, and it's not too common, lowering the dose work.